Okay, on slide number 8, you see a beautiful, colorful picture, and to the left in the upper left corner, we have a nucleus, and that's where all that transcription was happening. And we said in that surrounding that nucleus, we have a nuclear envelope. It's plasma membrane, but it has little holes in it called nuclear pores. And we said that DNA, those chromosomes are far too big to get out those little holes. But these messenger RNAs, these mature messenger RNAs, remember the ACBs and the AFEFs and whatever the other one was, they can leave. And when they get out into the cytoplasm, we said it's going to join up with those other kinds of RNA. First thing that's going to happen is we have this messenger RNA in the middle, and we have 2 pieces of ribosome that are going to come down and sort of squeeze it between them. And it's going to start sliding along that messenger RNA, and it's going to come to a point where there's a code that says start building that protein. It's going to be a start codon. And then what we're going to have is different kinds of transfer RNAs that are going to be complementary to the little words that are present. There are 3 base words that are called codons. So in your mature messenger RNA, we have all of these little words that consist of 3 bases that correspond to the different amino acids. So you got this RNA, look at your picture, this RNA sliding along, the ribosome, sliding along the messenger RNA, and it eventually comes to a place that we're going to call a start codon. And at that start codon, those transfer RNAs are going to start bringing in the different amino acids. And there's 2 different spots on that ribosome where transfer RNAs are going to sit, and you're going to produce those peptide bonds, join those amino acids together, and you're going to produce an amino acid that has the sequence of amino acids that was called for by the DNA. So we had said you had this code in the DNA, and then you copy that code into RNA, so you still got the code, and it goes out into the cytoplasm, and you have this ribosome sliding along it, and then at this point it says, oh, we need methionine, we need the first amino acid. And then it slides just a little further, and we bring in this second amino acid, and those 2 are joined together. And then it slides one more spot, and you bring in another one, and that one's attached to the previous 2, on and on and on, until when? Until you come to a stop codon. Stop codons don't have any transfer RNAs connected to them. So you go along, you're following the blueprint, you come to a stop codon, there's nothing more to be done. It all separates out, and it releases this new polypeptide chain. So remember, a polypeptide chain is just a sequence of amino acids that have been covalently bonded together with what we called a peptide bond. So we've got this primary structure of a protein put together. Now, what did we say? When we talked about proteins, we said they have a primary structure, and that's the sequence of amino acids, and that is directly coded in the DNA. And so we go out, I'm looking at my stuff here. So, all right, so I'm looking at the steps in translation, and so we had said that you find a start codon, and then you are sliding along, and when you reach the start codon, we're going to have a transfer RNA that's going to come in that is coded for in the RNA. And we have a picture on number 10 here. And you can see on the left, on slide 10, you can see what we draw. If I was to draw this on the board, I'd have drawn this thing that kind of reminiscent of a paperclip or something. And we draw. That's how people draw it on the board, because the right-hand picture is actually what it looks like. And who the heck can draw that? So anyway, so we had said on that messenger RNA, there is what we called a codon. And we had said it's a 3-base pair. It's a 3 bases that correspond to each of these amino acids. Previously, I mentioned that A's and T's are complementary and C's and G's. Now that's DNA. So and then we had said, yeah, with RNA, they have A, C, and G, but they have a U instead of a T. So we made a copy of that DNA using the language of RNA. So everywhere in the copy that you would have put a T, if it was DNA, you put a U. So we have these words that are made up of A, U, C, and G, and each word is 3 of these. And each one of these, you can go to a reference and see what each of these codons, which amino acid they code for. Now, we'd said the thing that's bringing that amino acid is the transfer RNA. And now it has the RNA that is complementary to the codon. So the messenger RNA has these 3-letter words called codons, and then the transfer RNA has a complement to that. It's not called a codon, it's called an anticodon. And so if you have the anticodon that corresponds to the word that's calling for methionine, you stick together, and that's how it brings in the correct amino acid. The transfer RNA has a anticodon that is complementary to the codon that's on the messenger RNA. And where did the codon come from? The genetic code that's in the DNA. So don't confuse those. All right, so you keep bringing these things in and building this polypeptide chain. And it has the sequence of amino acids that it was called for in the genetic code because of transcription and now translation. Now, when I mentioned ribosomes before, I said that they're little organelles that are bubbling around out there in the cytoplasm. Now, they're greedy little buggers. It ends up, if you look at the next slide, they don't like to share, and they don't really share, so we end up with what we call a polyribosome. So if you think of a messenger RNA, and we'd said the 2 halves of the ribosome come and jump on it, and it starts sliding down it, well, pretty soon, another one jumps on it, and it starts sliding down it, and another one, and it starts sliding down it. And as they go down that messenger RNA, we said each one of those ribosomes is going to start building that protein. We have a really cool photograph. I want you to look at it. It is slide number 11, labeled polyribosomes. And so what you see is some dots. You see some balls, and you can kind of follow the big balls. We see 3 of them, and then 1, and then 2, and then 5, and so on. And if you look carefully at that, each of those balls is a ribosome. And it's sliding along the messenger RNA and extending away from each of those ribosomes. It's a protein. So simultaneously, that messenger RNA, that mature messenger RNA, is simultaneously being translated, proteins are going to be built on it by multiple ribosomes simultaneously. It's what we call a polyribosome. And what does it take? It says it takes 20 seconds for each of those ribosomes to build a 400 amino acid polypeptide. And if you look at how much translation is going on at any given time, per second, 150,000 proteins are being translated per second. Oh, there we go, another 150,000, another, lots and lots. So in each cell, in every cell, and you have trillions of cells, by the way. Okay, I better stop right here. So we have reached a point where we have protein. And now, but we have the primary sequence of the protein. So we need to think about how do we get, remember we said there's secondary, tertiary, and quaternary sequence, structure to proteins. And what were they? That's the different three-dimensional shapes. And so we're going to talk about that.